Thank you to the organizers and the Sophion team for putting together this wonderful symposium. So my lab studies mechanotransduction, which is the process of converting biologically relevant physical forces like mechanical indentation, stretch, and shear stress into biological signals. Now, this process can occur through soluble proteins, membrane proteins, and other receptors, but my lab is interested in mechanically activated ion channels that are membrane proteins. So when there's mechanical force that's applied to the membrane or there's a change in membrane tension, these proteins undergo a conformational change and allow ions to flow into the cell. So if they're present in neuronal cells, then influx of sodium can cause a change in membrane potential, and there's a signal that originates. In non-neuronal cell types, calcium influx can also initiate the process. So classically, in vivo roles for mechanosensation or mechanically activated ion channels is in somatosensation. So Miriam gave a wonderful and beautiful introduction to what our sense of touch can do. You heard about proprioception from Thian yesterday, and then Steve also talked a little about this. So, and then John introduced us to dorsal root ganglion neurons. So the somatosensory neurons is what gives us the sense of touch in acute pain. There are three different types of ganglion: trigeminal, nodose, and dorsal root ganglion. And these neurons innervate the head, face, and neck. There's some innervation that goes to the internal organs, and then there are a lot of innervation that goes to the skin. So this has been known for a long time that mechanically activated ion channels are present in these somatosensory neurons and give us the sensation of external and internal senses. But then there's been precedence for cellular mechanosensation also, in the sense that tissue stiffness can change how quickly or how much a cell can migrate. There's traction force, so there's internal force that occurs within a cell that can also affect its biology. And then there's also evidence that tissue stiffness around the axon, for example, can guide axonal guidance. So my lab tries to understand some of these processes, and our favorite channel is this family that's called as the OSCA TMEM63A family. So these are bona fide mechanically activated ion channels that are conserved in plants and in animals. The gene name for plants is called as OSCAs, and there are 15 members within plants. In animals, the gene name is called as TMEM63. So Drosophila has just one family member, and then all vertebrates have three different types of family members. So they're conserved in zebrafish, mouse, and humans. So we identified this gene in around 2018, and prior to that, it had been shown that in plants, OSCAs was actually required for proper leaf and root development, and they were originally characterized as hyperosmolarity activated ion channels. And because we knew that osmotic shock can also change the mechanical stress on the membrane, we got interested in trying to think about them as mechanically activated ion channels. So since their discovery, now there have been precedents that there have been reports of human mutations that cause either hypomyelinating leukodystrophies, severe onset of developmental and epileptic encephalopathy, and TMEM63 for hereditary spastic paraplegia. In mice, there's been some evidence of chronic pain amputation and osmosensors and outer hair cells. And then in flies, there's been evidence that it's required for gustatory texture determination, hygrosensation, and lysosomal fat body morphology. So for those who are interested, there have been a lot of structures from different family members for OSCAs and TMEM63s. OSCAs are dimers, and TMEM63s are monomers from what we know. It's interesting in a way that each subunit encompasses its own pore, and 4 transmembrane domains come together to form an individual pore. So what we got interested in was that initially after its discovery, there were some reports of how mutations in TMEM63A causes hypomyelinating leukodystrophies. So there were some patients, there are about 12 to 15 now that have at least been reported that present with nystagmus, ataxia, delayed motor development, and hypomyelination of the deep and subcortical white matter. So what you see on the left are just serial MRIs of patients, either control or patients that are usually at the age of 3 months to 6 months. And what they had noticed, or what the clinicians had noticed, was that by the time they're at the age of 10, the myelination or the hypomyelination that was observed in MRI through MRIs at the age of 3 months had somewhat kind of compensated or caught on. So we kind of got interested that what is this mechanosensitive channel doing in myelination? And when we looked at the expression, it turns out that 63A is highly expressed in oligodendrocytes. So just to make sure that we're all on the same page, oligodendrocytes are the myelin-forming cells in the central nervous system. They originate as oligodendrocyte precursor cells that undergo some significant morphological changes as they differentiate into pre-myelinating oligodendrocytes and then mature oligodendrocytes. And when I went back into the literature and started to look at what can mechanical cues and mechanosensation do for this process, turns out that tissue stiffness can affect how quickly or how much OPCs or oligodendrocyte precursor cells can differentiate into oligodendrocytes. Stretch from processes as they look and search for axons can also cause calcium influx, so there's mechanical cues that originated there. Axon curvature also acts as a sensor or as a stimulus for the oligodendrocyte to know whether it should start wrapping the axon or not. And then finally, when there's myelin compaction, there's a great amount of hydrostatic pressure that occurs in the inner trunk. But I think the most fascinating clue that we got was that if one purifies OPCs and cultures them onto nanofibers, so there's no activity or there's no biological signal that's coming from the nanofiber, the oligodendrocytes can still wrap nanofibers and can maintain their axonal diameter selection. So for us, this meant that maybe there is some kind of a cue or biophysical cue that the oligodendrocyte can sense and allow myelination. So for the rest of the talk, I'm going to talk about how we think about 63A as a mechanosensor in myelination. This project is led by a postdoc, Julia, and then 2 grad students, Amanda and Tony, and then Sage Berryman, who's an RA in the lab. Amanda is also co-mentored with Kelly Monk, who's a myelin person at the Vollum Institute, and we'll talk about how their collaborations have helped us. So the first thing we did was what my lab knew how to do very well, which is to overexpress these genes in piezo-1 knockout cells and then look for stretch-activated currents. So what you see on the left are representative traces. This is in the cell-attached patch clamp mode. Steve introduced us to this technique yesterday. We can apply negative pressure in the cell-attached patch clamp mode, and when we apply pressure, you distend the membrane, and that's how we activate stretch-activated currents. So we see that as we increase the pressure within the recording pipette, we get more and more response or current. When we tested a couple of the previously reported mutants, turns out that these mutants don't produce any currents. So if we characterize just the maximal current that can be induced with an overexpression system, we see that most of the mutations, at least at that point when we tried, do not induce currents. So whether these mutations, so this is just a pictorial representation on the top right corner of the different transmembrane domains and where the regions of the mutations are. So because a lot of channelopathies are associated with trafficking deficits and not necessarily because it's a Dud channel, we tested this hypothesis by adding a bungarotoxin binding site on the extracellular loop of the channel and then binding with bungarotoxin that's conjugated to GFP and then take it into flow cytometry to quantify whether the channel of interest is going to the membrane or not. So our plasmid is in an M-cherry vector. So when we do this across an x-axis that's looking at M-cherry fluorescence and y-axis that's looking at GFP, we see that in untransfected cells, you're looking for the top right quadrant. There's no cells that are in that quadrant. When we have wild type 63A only transfected cells, you see most of the fluorescence on the M-cherry axis. But then if you do this in a plasmid that has the BBS binding site, you see this nice correlation between the M-cherry and the GFP signal. So when we went ahead and quantified this for either untransfected, wild type, and all the mutants, what we observed that most of the mutants were actually not making it to the membrane, so suggesting that these were trafficking deficits. To make sure that there was no premature degradation, we also looked at protein levels. So protein levels for these mutants were all there, but it's just that they're not trafficking to the membrane. So knowing that this was a loss of function mutation, and that loss of function of 63A can lead to hypomyelination, we wanted to test this on another model system. So we turned to mice. We had a global knockout mice. Now, if you remember, I mentioned that with the human patients, the diagnosis was usually done around 3 months. They see delayed myelination or reduced myelination from 3 months to up to 3-4 years of age, and then by the time they're 10 to 12, the myelination has caught up. So what we decided to do was go through a whole developmental window for mice and look for myelin coverage. So we can do this by staining for myelin basic protein. What you're looking at are images in the cortex of the mouse brain. This is at P14. You see nice tracks that represent myelin. And this is a wild type animal. And then we went ahead and did this with a global TMEN63A knockout. And then did this around 4 developmental time points. And what we saw was that at P14, we see about 40% reduction in myelin. We also looked at sheath lengths, and it turns out that the sheath length is actually the same. We also tried to see, or we also quantified whether OPCs were just not differentiating into oligodendrocytes, and that's why we were seeing less myelin coverage, and that's not the case between the wild type and the knockout. To test whether this is specific to oligodendrocytes, we generated a conditional knockout using a MOP-CRE. So MOP-CRE turns on only in mature oligodendrocytes. So again, these are just micrographs of a wild type and a conditional knockout. And similar to, and we did this only at P14 because we wanted to, again, pick the developmental window where we had seen the deficit in the previous mouse model. We see a 40% reduction in MBP coverage or less myelin without any change in sheath length. So, and we've tested this out in 2 different CRE lines. So we then turn to see whether it's actually conserved in vertebrates, and the zebrafish is actually a really nice model to look at myelination because they're transparent at a larval stage. So you can do in vivo imaging while these cells can start myelinating. So what we did was we took a MBP EGFP, and the way these experiments are done is in a single cell stage, we inject Cas9 and guides against TMEM63A, and then see whether myelin, using GFP signal, whether there's less myelination in the dorsal and the ventral spinal cord of these animals. So again, you're just looking at controls for Cas9 injected animals at 4 days and 5 days post-fertilization. And then when we compare this to the 63A mutants, we see less myelin only at 4 days, both on the dorsal and the ventral side. So this is again consistent with what we had seen with the mice and also consistent with what's been reported for humans. So at a cellular level, to see whether it's just that these oligodendrocytes are not making enough myelin, we turned to a culture system, which was initially characterized or standardized by Ben Emery in Ben Barris's lab. What we do is we take mice at P6 to P8 age, dissect out the brain, immunopan to get rid of the astrocytes and microglia, and just select for OPCs, and then culture them either on cover slips or on nanofibers. So when we do this on cover slips, you see these, they have this really beautiful morphology. MBP is again a representation of myelin, and then effectin is just to give you the margin of the cell. With the knockout animals, again, we see less myelin that's being produced by individual oligodendrocytes. So this was all good. I was super curious to see whether now, if we take away the biological aspect of this and just plate them onto nanofibers, do we still see the same phenotype that we would expect? And also, these cells are not meant to be on flat cover slips. So it makes more sense to try this in a more 3D system So we cultured cells either from wild-type TMEM63 or nanofibers, or from the knockout cells. And what we see is that in the knockout oligodendrocytes, they have fewer sheets per oligodendrocytes. So again, suggesting that not only is the cell making less myelin, but then it's also failing to recognize its mechanical cue in a way that it can wrap these fibers, or axons for that matter, adequately. So during the process of this project, there was some evidence, there was a lot of evidence coming up that these channels were actually present on lysosomes. And Lily Jan's group here had shown that they were present in Drosophila, they're present in lysosomal fat body, and is required for morphology. So to see whether this was true for an endogenous system in mice, we took brain extracts from wild-type and knockout animals. And then because there were no antibodies, we used good antibodies for tissue. We used Westerns to see if we did a fractionation experiment of trying to regulate or trying to separate out different organelles and different regions of the cell. Where does TMEM63A really localize? So LAMP2 is used as a signal to look for lysosomes. MBP is again myelin, and then TMEM63A is just the lane for the protein by itself. And what we find is actually TMEM63A co-localizes both in lysosomal compartments as well as in the plasma membrane compartment. So we think that it's actually present in both places. The other thing I want to bring your attention to is these experiments were done in animals that were at P30. When we did immunofluorescence, it looked like at P30, we were, the myelin had completely caught up. But during using biochemistry, we're still seeing reduced myelin in P30 animals. So we think that it doesn't completely recover, and it probably persists on for a longer duration. So knowing that now it's present on lysosomes and plasma membrane, there was quite some evidence about how lysosomal trafficking can affect myelination. So we kind of got interested in knowing whether the channel is present at the plasma membrane always, or is it constantly getting trafficked and recycled between the plasma membrane and lysosome. So we went back to our bungrotoxin binding site, or bungrotoxin binding site in our TMEM63A, and did sort of the cycling or time-lapsed imaging in a way that you would call it. So these are HeLa cells that are overexpressed with TMEM63A, just wild-type TMEM63A that has the bungrotoxin binding site. And so M-cherry again is for transfected cells. What you see on the left is just a control where there's no bungrotoxin added to the, when there's no bungrotoxin added to the cells, either at 0 hours and 2 hours post-addition. So at 0 hours, you see that when there is bungrotoxin in the media, you see nice punctate staining indicating that the channel is present at the plasma membrane. But 2 hours post that signal is completely gone, suggesting that the channel is actually probably getting internalized in a live, at a dynamic pace. So this is just the quantification to show that at 0 hours, in the presence of bungrotoxin, we see nice intensity at the plasma membrane, but that signal is gone 2 hours after it. As a control, we did this for piezos just to show that this was unique for TMEM63A and not for any channel. If you look at just the micrographs on the right-hand side, we switched to hex cells because piezos prefer hex cells. We see staining at 0 hours from bungrotoxin for piezo2, and then 2 hours later that the signal still remains, suggesting that the effect that we're seeing for TMEM63A is more unique. So to sort of summarize everything that I've showed to you today, TMEM63A is the mechanosensor that drives myelination in oligodendrocytes. It's conserved across multiple organisms. It's associated with this HLD19 or hypomyelinating leukodystrophy. And then we're now starting to think about what are the downstream signaling mechanisms of a mechanosensor that's required for myelination. So we think that with its presence as a tension sensor at the plasma membrane, it's probably required for axonal selection and gives the cue to the oligodendrocyte to start wrapping. Its presence on lysosomes and plasma membranes is also interesting because a lot of lysosomal storage disorders also cause hypomyelination. And then there's been evidence that these channels, although are non-selective cation channels, could also flip lipids, which is again a very intriguing process for a cell that undergoes a large amount of lipid biogenesis. And in the long term, we'd like to use the zebrafish as a model system to screen for drug targets that can remediate hypomyelination. So in the end, I acknowledged all the people who did the work, but I also want to thank the OHSU Flow Core and the Imaging Core and the DCM staff that helps us with our mice, my funding sources, and our collaborators, Kelly Monk and Ben Emery at OHSU. So with that, I'll take questions. Thank you.
Okay, that was great.
Thank you.
I may have missed this. Does the phenotype you saw in the mouse model, if that's transient and then myelination kind of returns, is that true in all of the patients?
Yes, yes. So I think, so the patients are probably in their 30s now. They got diagnosed a while ago. So we don't know if, it compensates, so there's recovery by 30 and then with age, whether it changes or not. So we're aging some of our mice to 1 year and then trying to see with the, at an older age, whether it reassociates or represents itself.
I had a really similar question, but I was just curious why you listed the deficits at this really early time point. Does 63A expression decrease
with age? Actually, we haven't tried, but it turns on only in myelinating oligodendrocytes. So mature myelinating oligodendrocytes. So there's something about the way that the cell, when the cell is starting to look for its axons and start myelinating, I think that's where we see the strongest phenotype. And then after that, there are other compensatory roles that then kick in. So myelination, it looks like they're all minor deficits, but anything stronger than that, the animal will die because evolutionarily is such an important process. There's also piezo2 is expressed after somatosensory neurons. Oligodendrocytes is the next cell type where piezo2 is very highly expressed. So we're now starting to do piezo2 63A individual and double knockouts to see if there's compensation from other mechanochannels.
in places with like a lot of neurogenesis, like the olfactory place?
Yeah.
Would you expect that this is more active throughout lifetime? Because there's always more.
That's a good point. Yeah, we haven't thought of it. We've looked in the optic nerve, just because that's where myelination first starts before it starts in the brain, and we've tried to look at numbers of oligodendrocytes versus OPCs. But yeah, we haven't looked in the olfactory bulb or other places where there's more increased activity.
That's it. I don't want to stand